Symbioses, the interactions between two different species, make the world go round. Everything from our agricultural systems to our own gut depends on groups of interacting organisms doing their thing. Our guest today, Dr. Allison Hansen, studies how a common aphid gets its nutrition. It turns out that its entire diet is dependent on a bacterium living inside each and every aphid. But how did it get there? What does it do for the aphid? Let's dive into the big world inside small insects. This is Radiobio.
Don't know much biology.
Hi everyone, welcome to Radiobio. I'm Jeff.
I'm Jasper.
Today we're joined by Dr. Allison Hansen, professor of entomology at the University of California, Riverside.
Hello.
Hey, can you tell us a little bit about yourself and what you work on?
So I work on insect-microbe interactions, and I primarily work with microbes that are specialized inside of these insect cells. But I'm really interested in the microbiome as well with insects. So I have a variety of microbe-insect interaction projects, as well as kind of plant pathogen interactions with those microbes. So kind of a diversity of interactions.
So how did you get started, and why did you decide to study the symbionts and their hosts?
That's a great question. Well, I started out, I really liked nature, and so I didn't really know what I wanted to go into, but I was always fascinated by different species interactions since I was little. And so I really liked plants and animal. It was just a very general. And so I started out in forestry, actually, and then really got pulled in forest insect interactions and learning, how do trees die and things like that. And then I just got really more sucked into the genetics and the evolution of these systems. And so I actually started out in community ecology more for my master's, and then I just completely transitioned more to the molecular level because I just kept on asking questions of, okay, how does that work? How do they interact?
And so what kind of insects and microbes are you specifically focused on?
So I like to study the model, to study insect-microbe interactions, and that's one of the most intimate obligate associations. It's kind of a classic story of symbioses, particularly nutritional symbioses. And so these are called hemipteran insects. And so some of these hemipterans feed on sap. And so sap is very high in sugar, so it's a very sugar-rich diet, but it's very low in nitrogen, particularly essential amino acids. And so, like you, me, we all need these essentials. We could eat a hamburger, soy, whatever you want, but, insects don't have that option, particularly these sap-feeding insects. So they have to somehow get these essential amino acids. And so they're able to do this by having this association with these microbes. And so these microbes provide these essential amino acids, provision them, so they can feed on this diet that is just so high in sugar, but low in nitrogen. So all sap-feeding insects need this some type of obligate symbiont to provide these nutrients. So that's why it's kind of a classic model. The insect can't survive without it, and insects have diversified through evolutionary time with their symbiont and co-speciated with them. So it's this wonderful evolutionary innovation, this partnership between other domain of life that basically allows them to feed on this diet. So it's kind of an interesting interaction. I'm like, how does this happen? How does this metabolism work together? Mm-hmm. Since they had so many, millions of years of evolving together.
Dr. Hansen mentioned obligate symbiosis, which are examples of mutualism, where the symbionts are obligated to be in the interaction to survive. In this case, we're discussing the microbes inside aphids that need the host to survive, and in return, they help the insects get nutrients from their food.
And do we know kind of roughly what proportion of, let's say, the hemipterans have these symbionts? Is it all sap-feeding hemipterans, or is it most of them? Is it only a few of them?
For the most part, in general, it's most hemipterans need this if they feed on a sap diet. When you include other insects that need obligate symbionts, that's about 10% of insect diversity, which is a pretty big chunk.
That's huge, yeah.
Considering insects are one of the most diverse animals on the planet. So, yeah.
When she says 10%, this is a huge amount of insects. It's estimated that there are 2 to 30 million species of insects on the planet, which means there are roughly 200,000 to 3 million insects that interact with symbionts.
That's a lot. Well, how did these bacteria get into these insects? We asked Dr. Hansen what she thought.
Yeah, I mean, there's no direct idea of how it could have been gut and then worked its way in, or whether it was a pathogen, or if it was a plant pathogen, then it's going to be drinking, plant sap, and then the pathogen's going to get, sometimes the pathogens go through the salivary glands, actually go through the insect, through the hemolymph, but they're not symbionts quite yet. But there is this interesting intersection where I think some plant pathogens can have that interface between being a symbiont but also a plant pathogen, and that's kind of an area I'm fascinating in too.
And so once these symbionts are in these things, I mean, they are, that's actually one question, is how do they even get into the host in terms of life cycle? Are they born with these symbionts? Are they passed down? Do they grow with them?
So the intimate symbioses I'm talking about, they're maternally transmitted. So mom gives, to offspring. And so it's kind of the same way to think about mitochondria, really.
Okay, so the symbionts are maternally transmitted like mitochondria, the powerhouse of the cell. But the difference is that as an obligate symbiont, its only function is to benefit the host, and because of that, its genome has retained genes that only serve this purpose.
Because they've lost the other ones. When you don't use it, you lose it. It's relaxed kind of selection. So you could really tell what kind of the function is just by reading the genomes, of these symbionts, because of the way the genomes evolve. So through omics approaches like genome sequencing, kind of approaches to look at the microbe sides and then also the host sides and how they interact with these. So we isolate the special cells that hold the symbionts so we can tell symbiont specific kind of gene expression with the host as well as the symbiont, and compare that to other host tissues that don't have the microbes in it. So you can kind of do a comparison.
And what are those specialized cells that hold the symbionts? What are those called?
They're called bacteriocytes.
And where are they found in these hosts?
So they are in the bodies, kind of near kind of the gut layers. So not in the gut, but they could be surrounding them in various types of hemipteran insects, for example. So kind of in the body, floating in the insect blood, also known as hemolymph. So.
Okay, so these symbionts are maternally transmitted and live in these specialized cells in the host and have small genomes whose only purpose is to serve the host. Sometimes in these types of symbiotic relationships, there can be horizontal gene transfer, where genetic information can be directly transferred between individuals. So we wondered, how do these symbionts use horizontal gene transfer, or do they communicate with their host? So we asked.
They actually lost some of the genes that allow them to do horizontal gene transfer and to recombine. So they're just constantly losing genes and not getting really any novelty as far as new gene.
There's no gain.
No. And so in certain insects, they actually have more than one obligate symbiont. So they're not fully replaced in those situations, but they have companion symbionts. And when they sequence the genomes, what some of these scientists that study them find is that they lose certain pathways where the other symbiont retains them, and there's this perfect puzzle-like complementarity of pathways. It's really cool, to see that pattern of just complementation of that. So
it's like a symbiosis between the symbionts within the host that they are also
right. And it's a puzzle of how exactly those symbionts interact, and then the host on top of that. So a lot of scientists in the field are just trying to figure out the basic regulation of these different organisms inside of it. It's like the Russian doll kind of.
Hey everybody, don't skip this part. This is huge. These organisms are only losing genes over time, and the genes that are left are only there to help the host. Once they lose enough genetic information, they get replaced by other symbionts, grooming or paving the way for the next one to fill in the slot.
That's so weird. What role does this microbe have in the host, let's say, getting nutrients that's missing from sap? How does this microbe kind of influence that process?
Okay. So primarily in the field, we think the host is in control of this metabolic kind of collaboration in general, animals cannot produce essential amino acids. We don't have the pathways. So it would be more like the enzymes are working in reverse. They have the end steps just to recycle these important essential amino acids into like glutamine or some immunodonor. But we're thinking maybe they work the other way to help. They adapted this way to produce them, to regulate those end steps.
That's so cool.
So through these different host control mechanisms, we're kind of thinking that's what's regulating the system. But then some of my research stumbled on maybe the microbe is doing some aspects of this regulation, especially in stages early in the embryo when it doesn't have this specialized host cell around it. And this might be regulated by small RNAs because it lost pretty much most of its regulatory proteins for bacteria compared to its free-living relatives. So
just as a refresher, small RNAs are involved in a whole bunch of regulatory processes and gene expression. And because they're small, they can operate at a fast rate.
So going back to the symbiont world, it's been found in small genomes that some of these small RNAs are actually expressed as well. And so it's just, is it noise or is it important? And so that's what my lab is, one project we're trying to figure out if these small RNAs are very important in the regulatory steps of facilitating gene expression.
So who would you say is driving the evolutionary bus?
That is the question. Well, I mean, in the field, the dogma is pretty much the host is in control of this relationship. The genome of the bacteria, it's being transmitted from mom to offspring, like I said. And so from these events, there's bottlenecking of events where only a few cells, inoculate into the embryos to be passed on. So from this, it's selection is not working strong enough in these cases. So it's argued that it's kind of a host-restricted microbes are, adaptation doesn't really work very much in these systems. So it's...
The selective pressure is more on the host than on the symbiont, because it's just on essentially the hosts that have offspring.
But we don't know fully yet of, both sides of the story. And so I think it's something to consider, though, like, sure, we've been thinking more of the adaptation from the host side and the control of this kind of domesticated relationship, but maybe we should start to think of these other aspects that maybe there is something going on also on the microbe side, some selfish interests as well, or things. So
it's really interesting to me. You keep saying domesticated relationship. I mean, it kind of makes sense. It's almost like these hosts are kind of like potentially kind of even corralling these symbionts and like using them in a way. And yes, it's a mutualism, but you said that their genomes are downsizing to the point where they sometimes are losing the ability to do other things.
Right.
So like what actual evolutionary benefit are these symbionts getting out of this relationship?
Yeah, I mean, you can argue that, they're becoming, if they can't recombine, they're becoming asexual. So it's just this kind of homogenation that occurs and limited diversity and adaptability. But I also, small RNAs they can evolve rapidly too from the RNA world of things. So going back to the RNA world when, protein is, not as, important yet for the regulatory, starting of life, maybe how did life start? So going back to that right here, you still have the ability to have small RNAs do some type of, maybe they're stepping in more and doing regulatory functions because they can't get new proteins, so it's a little bit harder, slower from that evolutionary process, but just something to consider.
And how does the ecology of the insect hosts come into play? So does, let's say, a plant that these insects are living on or the environment they're living in, can that influence this symbiosis in some way?
Yeah, and something I'm kind of interested in for my research direction, that different plants have different nutrient and defensive compounds. So how do these different, diets that they're feeding on, I mean, it's primarily sugar, but there's other stuff in there, and there's different levels of nutrients, and it varies even on one host plant, so it can be difficult.
Like where they are on the plant, what specific part they're
so not
eating.
only just the different species, but there's a lot of variation out there. So how does variation in diet affect the regulation of the symbioses? That's something that I am interested in. So one grad student just published a study showing that there is a difference in regulation of some key genes that are host genes that are very important in the symbioses, particularly transporter, and some genes important in recycling ammonia. So I think there might be potential host control of a compensatory kind of response, metabolic response, to try to get more nutrients when they're feeding on a poor nutrient diet.
So it's almost like it's the host is missing a nutrient, and that is actually upregulating something going on in the symbiont so that it can get something back from the symbiont.
Right.
That's really cool.
Yeah. So something we'd like to explore a little bit more in depth. And we're looking at different types of regulation too, because we're looking at epigenetics as well. And so we're finding that when they're feeding on different host plants, there's different methylation signatures in these bacteria sites. And so we're interested in finding out if that's linked to differential gene expression, like the fact that they can go out, feed on a plant, and some signal from the environment, nutrient, or host plant compound chemical can then turn on switches that can like change the regulation of that, which would be really cool. Mm-hmm. And so that's something we're also looking into deeper. So it's still like on the fringe, I think, of is it important or not, but that's kind of the epigenetics field in a lot of aspects of what you're studying. But could be something interesting as far as adaptation in the natural environment, especially since some of these insects are asexual or clonal during parts of their life stage. And so genetically, they're all the same out there, but they can maybe be influenced by epigenetics and have these polyphenisms being basically triggered by environmental cues, then differentiate, methylate their genomes, even though they have the same DNA blueprint, but that's a whole other level of regulation that's like, but it's really fascinating, cool. I think. Yeah. But so I guess my lab, I like to study these weird fringe areas of regulation of epigenetics, small RNAs, like, that sound cool. I don't know if they, but no, I mean, we're finding interesting patterns, so we keep on chasing them down, which I think that's the fun part of being a researcher, of finding, making sense of puzzles, what's real, what's not. So
this has been so cool to learn about these microbe and insect interactions. What roles do other symbioses play in our world? But
there's so many cool insect interactions to study, though, not just hemipterans, but with gut microbiomes and like mosquito stuff, or bees, or like all these very important insects that either important pollinators or important for disease kind of vector, and hemipterans are more important for agricultural reasons. So, all these things are important for basic science and understanding evolution and how it works in symbiosis. But on top of that, insects are very important as far as from an economic perspective. They're pests of our crops, our fiber,
Pollination.
Also, pollination, dying bees. So everyone's heard, I think, about that.
Gonna make everybody sad.
Yeah, why don't we talk about something happy, like aphids?
So when you're out, let's say just walking around town or anywhere on a hike or something, and you see aphids in a tree, do you instantly kind of think of their symbionts, or do you look at them as just aphids?
Oh, that's a good question. Well, I mean, I feel blind most of the time, so I'm just trying to see the insect first, and then I don't know. I guess I would have to get it and dissect it and not really get into it like that. I do think about dissecting insects a lot of the time, but yeah. That's the only way to really see, like, inside of them. I mean, these insects are so small to begin with, so that's the problem. But I wish they were bigger. I wish there was a way to, like, make myself become microscopic and I can, like, crawl into them and, like, just collect the material more easily and, like...
just grab your symbiont and walk
yeah,
out.
It would be so nice. And
bit of a Rick Moranis situation. Funny, I blew up the aphid.
Yeah.
So if you had like infinite funding, if you won the best NSF ever,
that wouldn't be an infinite funding.
Okay. Hypothetical insert funding situation here. What would you work on?
What would I work on?
Yeah.
Well,
What project would you take on?
probably the pet projects I have of just understanding more of the evolution of these, like especially the small RNA aspect. I think there's a lot of interesting things to kind of explore a little bit further, and also the epigenetic kind of stuff, the regulation of that. So it's NSF, that's more like basic science, so that's probably the areas I'd go. But I think there's a lot of cool applied kind of projects as well that would help, agricultural aspects too. So that would be a different funding agency.
Yeah.
What's kind of your sum up either favorite thing about your study system or something you wish everyone knew about aphids and symbionts and interactions?
Oh, man, that's a hard one. My favorite thing? Well, I think it's unfortunate that people don't like aphids as much as other insects. I think, I think people should realize that aphids are really awesome. And they are agricultural pests, so they're important to study. But yeah, I mean, they transmit a lot of plant diseases too. A lot of hemipterans do. Some are like citrus greening with the psyllid. That's like destroying citrus industries around the world. And so I think they're really important, like economically, and some people just don't realize because they get buried under everything else. Like, of course, like we care about human disease, vector diseases. Those are very important too. I'm on some mosquito microbiome projects. So I'm not going to throw the mosquitoes under the bus or anything. But yeah, I think, if people understand where their food comes from and how important sustainability of agriculture and just, understanding the importance of plant-eating insects. And aphids are one of those, but also they're just really cool insects. So they look like just, when people ask about aphids, there's like, are those things you could smush on my roses? And like, I wish maybe I need to like make them more charismatic some way. And I don't really know how to do that. But if anyone has ideas or...
It's a tough one.
Yeah.
I mean, you play up the fact that they carry these little
make
autonomous things inside of them.
There's those little pillow, fuzzy microbe animals and phages now. So maybe like make little cute pictures of the aphids hugging the little microbe or something.
Yeah, if they can make a toxoplasma pillow.
Yeah.
Yeah, so the next time you see an aphid on your houseplant, just take a moment to marvel at the little universe of autonomous microbes living inside them. This episode of Radiobio was brought to you by Jasper Toscani Field and Jeff Lauder. Produced by Jackie Shea. Edited by Jasper and Jeff. With artwork by Ann Deep.
Radiobio is produced by graduate students at the University of California, Merced. Support for Radiobio comes from the Quantitative and Systems Biology Graduate Group, the School of Natural Sciences, and the Graduate Division at UC Merced. You can help support Radiobio's mission of increasing scientific literacy in California's Central Valley and beyond by donating at giving.ucmerced.edu/radiobio. Find out more about our mission, events, and podcasts at www.radiobio.net.